In Chapter 3, we're going to be studying and looking at the cell, both the structure and function of the cell, which is the smallest living portion of a living organism. And so in this slide, we're going to go through each of these parts a little later in the chapter, but this slide shows you just a general schematic of what a cell might be like and some of the major parts, and we'll be going over each one of these a little later. But to begin with, section 1 just kind of briefly mentions a few of the functions of the cell. First of all, one of the main functions is to go through metabolism, and that is basically how the cell uses energy to survive, to live, and to have the building blocks that it needs to live. It also goes through various transport mechanisms, and we'll be talking about different chemicals such as proteins that help to allow for transport of different molecules across the cell membrane. Cells have the ability, and they go through communications. And so we'll be talking about how cells can go through communications. A lot of times that's through a chemical signal. And when you get to 169, you'll be talking about the endocrine system, and that uses a lot of chemicals to relay information from cell to cell. Other times it's an electrical signal, and so cells can transfer electrical inputs from one cell to the next. And then cells have to be able to reproduce, and we'll be talking about in this chapter one type of cell division. You probably already heard about this, but it's called mitosis, and so we'll be talking about the various stages of mitosis. Then in 169, you'll generally talk about meiosis, as it refers to cell division that you find in the reproductive system. So to understand the cell, we need to understand kind of the boundary of the cell, this plasma membrane or the cell membrane. And it's basically what is called a fluid mosaic model. This is the theory behind how this cell membrane works. It's fluid in the sense that it does kind of move around. And in the picture here, you see an example of a cell membrane, and it has embedded in the cell membrane different types of molecules. So there's some protein molecules that are here, there's some carbohydrate molecules that are here, cholesterol molecules that are kind of scattered throughout, and that this is fluid that these molecules can move around in that cell membrane. And it's a mosaic, which means it has different types of molecules embedded in it. And so one of the things that we look at when we look at cell membranes is the types of molecules you're going to find, which are lipids. And so one of the things to note here is that your cell membranes are made out of what's called a phospholipid bilayer. Bilayer just means two layers. And these are phospholipids. You have two layers of lipids that line every cell membrane that you have. And they have the element phosphorus in it. And so that's why it's called a phospholipid bilayer. It also contains cholesterol. And so in the picture here, you can kind of see some little yellow-like substances, kind of molecules kind of scattered throughout. That is a cholesterol which is needed in your cell membranes, and that helps to stabilize the structure of your cell membranes. Now, some researchers estimate that a human body would have trillions of cells in the body. Another chemical that you'll find, or another structural biomolecule that you would find, would be membrane-bound proteins. These are proteins, just like we've seen in chapter 2. These are proteins made out of amino acids, but they're embedded in the membrane. And they can either be integral, which means they're going to span across the entire bilayer, or they're peripheral, which means usually you would find these kind of on the inner surface here of the edge. We'll talk about 5 main categories of membrane proteins, beginning number 1 with transport proteins. Transport proteins do what they sound like. They are there to transport some kind of substance, a molecule, a chemical, an ion, across, back and forward, across that plasma membrane. And so here you see in the example here, you see a protein channel that allows for some type of molecule to go across here. And usually the channel proteins are usually open. In other words, they don't have a gate or any type of substance here. And that allows the molecules to move either across to go outside the cell or back into the cell, where you find what's called cytosol, or the cytoplasm of the cell. Sometimes transport proteins are carrier proteins, and so that means that an ion, a substance, a chemical is going to bind to it, and it causes that carrier protein to change shape. And when it changes shape, that kind of opens the other end of the protein and allows the molecule to pass all the way through. So we'll see this again a little later when we talk about the energy requirements to get some of these actions to take place. Number 2 are receptors. Some proteins are receptors. Some membrane proteins are receptors. And by definition, a receptor is something that's going to bind to some kind of chemical. And that chemical messenger a lot of times is called a ligand. That's just a vocabulary word. And so in the example here, you see some kind of protein receptor. And you'll see more of this when you get to the endocrine system, because this is how your endocrine hormones work. You have some kind of hormone maybe here, or in this case, it's just called a ligand. It binds to the receptor, and that kind of opens the door, opens the gate for this protein channel. And that allows molecules to go through that channel. It's like a tunnel system through the membrane. Some proteins work as enzymes. Enzymes are basically catalysts, biological catalysts that help to speed up reactions. And so some enzymes that you have are bound as proteins, as an integral type protein in the membrane. And so they can actually work to speed up a reaction where they're located, there inside the membrane. Some proteins are there to just give structural support. And so they help to maintain the shape of a cell and its integrity, so that the cell doesn't collapse. It has some certain types of protein filaments inside the cell that kind of makes the cell stronger. And we'll talk a little bit more about those types of proteins. There's something called a cytoskeleton we'll see in the second part of this chapter video. Then some proteins work to attach cells together. And so these are attachment proteins. Sometimes they're referred to as linker proteins, and they help to attach certain types of cells together. You see this a lot in the heart, the cells that make up the heart, because they are linked together, and that allows cells to communicate very easily different signals back and forth across one cell to the next cell. Now, section 3 talks about transporting. Now that we kind of understand that you've got a cell, you've got a cell membrane, you've got the different proteins that do different functions, now we need to figure out how cells send signals or chemicals or substances or whatever back and forth across that plasma membrane. That's called transport. And so one thing to note is that the phospholipid bilayer, or the cell membrane, that plasma membrane, is selectively permeable. You may have seen this word before. And so that it means it allows certain things to pass through, but it prohibits other molecules. So it selects, in a sense, what can pass through that membrane and what doesn't pass through. And so we'll talk about different ways that substances or chemicals pass through. Many times these are passive type mechanisms. Passive means that it doesn't need any kind of energy. It just happens by what's called diffusion. And we'll define that in just a minute. Other systems do require energy, and so they're called active transport. It takes energy for some molecules to pass across the cell membrane. So let's look at selective permeability a little bit more. There are 3 variables that help to determine whether or not a plasma membrane is going to need, or a particular substance will be transported by a passive mechanism versus an active mechanism. And so 3 things. Number 1, it depends on the type of substance. Certain substances pass very easily through a membrane, some do not. And so many times it depends on the size of the substance or the charge associated with that particular ion or substance, whether that substance is lipid soluble, will it dissolve through the membrane, or is it water soluble? Is it going to have a hard time going through the membrane? And that kind of leads to the next one, plasma permeability to the substance. How permeable is the plasma membrane to that particular substance that you're trying to transport? If it's a lipid, that usually transports very easily through the membrane. But if it's a molecule that is water soluble, maybe it's a protein, or maybe it's a certain type of ion, or whatever, or even water, those have to have some type of protein carrier channel to help go through the membrane. And also the concentration, how much of that substance do you have? That is also a determining factor of how you're going to transport something across a cell membrane. So let's look at passive transport first. I've got a couple of definitions for you. Diffusion, and we'll look at simple diffusion and facilitated diffusion. We'll also talk about osmosis. So here's your definition for diffusion, is moving some type of molecule from an area of high concentration to an area of low concentration, right? And so if you don't know this word, a solute, this is what you would dissolve into a liquid, right? And so if you're talking about a cup of salt water or a beaker of salt water, right? The water is the solvent, the salt would be the solute, and you mix those together and you would have a solution. Well, the solute molecules, or whatever molecule you're trying to move, if it goes from high to low concentration, that's diffusion. That's a natural process. In other words, that's passive. It doesn't require energy. Usually molecules want to go from high concentration to low concentration. So in the diagram, they kind of give you a little example. They've got a beaker here with a divider, and they've got some kind of molecule over here. They say it's glucose. You've got a lot of glucose molecules on one side. And so when you take that divider out and you only have a few molecules on the other side, you take the divider out, it wants to naturally move, the glucose wants to naturally move from a high concentration of glucose to a low concentration until it reaches a balance or equilibrium. That's passive transport. This is just another example of passive transport. You see sometimes passive transports will have some type of channel protein just because they don't diffuse directly across the membrane. Notice up here, where it says number 3, lipid-soluble molecules, they can diffuse directly through the membrane. If they will dissolve in a lipid, they can dissolve through the phospholipid membrane and go directly through. If they won't dissolve, then they may need a channel there so that they can fit through and go through the membrane. But this is still diffusion. This is still going from high concentration to low concentration. Remember we said that the membrane is selectively permeable, so there are some molecules that may not be able to go through the membrane at all. Sometimes you see the word facilitated diffusion. The word facilitate means to help, and so this is still diffusion. It's still passive, but these molecules have to have help, and that's from some type of a protein channel. And so that's why in the pictures here you have some protein channels embedded, and so these ions here need help going across. They're still going from high concentration to low concentration. Maybe they're moving in, maybe they're moving out, but they always go from high to low concentration, but they need help from some kind of protein channel. That's facilitated diffusion. Osmosis is also diffusion, but in this case, it's the diffusion of water. And so water also goes through the same type of movement. It's going to diffuse through a membrane, but notice that it goes toward the area where there is more dissolved solute. Right? This is still a passive process. There's no energy needed, but notice here in the definition, water moves from an area with lower concentration of solute, which means essentially more water molecules, across the membrane to the area with higher concentration of solute. So it starts out here. You've got a scenario here where you have more water on the outside of the membrane, less water here. And so water moves by osmosis from an area where there's more water to less water. Or you can think of it in terms of the solute. Water moves in an area where there's less solute to an area where there's more solute. And that's because of something called osmotic pressure. The more solutes you have, that creates more osmotic pressure, and it essentially draws in the water toward the solute. And eventually, you'll reach some kind of equilibrium, which means that you basically have balanced out the amount of water and solute on both sides of the membrane. In this diagram, you see a couple of terms up here: hypotonic, isotonic, and hypertonic. Hypo means lower than or less than. And so a hypotonic solution has a lower concentration compared to what's on the other side of the membrane. Iso means equal. So isotonic solution has the same concentration as what's on the other side of the membrane. And hypertonic means more than. And so, for example, you could take a red blood cell. The blood cell membrane would be your selectively permeable membrane. If you would put a red blood cell inside of a beaker of just plain water, then that would be a hypotonic solution compared to what's inside the red blood cell. In other words, there's less solute in the water than there would be inside of here. So water wants to go into the cell because water goes from a high concentration of water to a low concentration of water inside the cell. Or you can think of it again as going from a low solute concentration to a higher solute concentration inside the cell. And so that causes the cell to expand. If it's in an isotonic solution, those are equal concentrations on both sides, and so water just kind of will balance itself out. It's an equilibrium type scenario. For hypertonic, you put a red blood cell into a hypertonic that has a higher concentration of solutes dissolved, and so water goes from an area, again, of low solute concentration toward the area of higher solute concentration. It wants to go toward the solute, and so that causes that cell to shrink. Now, water can either go through the membrane, but because the membrane is lipid soluble, or basically a lipid, usually there's a protein channel just for water, and that protein channel is called an aquaporin. That's the protein channel that allows water to pass through a membrane. You may have a small amount of water going through that phospholipid layer, but if you have a situation where you need water to pass through very easily, such as the kidneys and the renal tubules inside of the kidneys, you need many of these aquaporins to allow water to move across the membrane. Now, the other topic here is the active transport mechanisms. Remember, active transport requires energy, and the energy that it's going to use here is called ATP. And that provides the energy for molecules to go against a concentration gradient. So in other words, they're going from an area of low concentration to high concentration. So active transport is opposite of diffusion. It's going from a low concentration to a high concentration. That's going against the gradient. And we'll talk about that there's two different types. There's primary and secondary active transport that uses certain types of carrier proteins as basically pump mechanisms to try to set up a concentration gradient so that they can move across the membrane. Now, these pumps come in three different categories, and they're basically little vocabulary words here. You just need to memorize the prefix here, uni, that means one. So a uniport is a type of protein pump that is pumping through active transport just one type of substance across the membrane, either going into or out of the membrane or out of the cell. Symport means the same. And so here you're pumping or moving two or more substances through the membrane, but they're all going in the same direction. So you're pumping two molecules of sodium at the same time, going out of the cell membrane. Two molecules coming into the cell. Antiport, you got two or more molecules being transported, but anti means opposite. And so you're going in opposite directions. One's going in, one's going out. So these are some diagrams from your textbook. Primary active transport is showing you some diagrams here. Number one, here's your protein pump that's embedded in the membrane. So this is an integral type protein embedded in the cell membrane or the plasma membrane. Here you have sodium ions, and notice that the idea is you're trying to move sodium ions against the concentration gradient. So inside the cell you have fewer sodium, outside the cell you have more sodium, but you're trying to move more across the cell. And so what will happen is some of the sodium ions fit here. Then if you follow it to step two, it uses energy, ATP energy, and that causes this protein to change shapes, and that kind of opens the door. You have to have the energy to get the protein to change shape. That opens the door on the other end. That allows the sodium, it kind of transports and delivers that sodium outside the cell, and then at the same time it picks up potassium. Again, that potassium wants to go against its concentration gradient, and then whenever this embedded protein reconfigures its shape, then it opens up the other end, and that allows the potassium, in this case, the K here, the potassium, to go inside the cell. And this process repeats like clockwork to try to move sodium against this gradient and moves potassium again against this gradient. Secondary active transport has an indirect way that it uses ATP. And so this is really a two-step process. I have step one listed here. ATP is first used to generate a concentration gradient. And so what ATP is used, just like we said before, you've got a sodium-potassium pump. You got sodium on one side. The energy is used to pump out sodium outside the cell. So now you've got a lot of sodium sitting outside the cell. That's a concentration gradient. You're establishing a gradient with more sodium on the outside and less sodium on the inside. Step two is now that you've got this concentration gradient, you can use a different type of a carrier protein. This is not a pump, so this doesn't use energy. But now you've got all of this extra sodium out here. The way this carrier protein works is notice that it's a symport. It's going to move two molecules the same direction. And so here you got a lot of the sodium out here. The sodium binds, and when the sodium binds, it kind of pulls with it another substance, glucose in this case, in the example. And that concentration gradient is what's driving the transfer of sodium and glucose from this side of the molecule, from the outside of the molecule to the inside of the molecule. So this step doesn't use energy. That's the indirect portion of it. That's the secondary portion of it. It does require energy to set up the concentration gradient, but once you have the concentration gradient set up, it doesn't require energy now to move the glucose because this is a symport. It moves two molecules, same direction. There's other ways to actively transport molecules or substances back and forth across the membrane, but this uses vesicles or storage containers. And so there's a process called endocytosis. Endo means taking in, inside of. Cyte is a root word meaning cell. So endocytosis is drawing in something, and one way that cells do this is a process called phagocytosis. That's cell eating, where a cell basically wraps itself around some kind of particle. You see this in certain types of white blood cells where they're part of your immune system is trying to get rid of some kind of bacterial or some type of dead cell or broken debris type cell inside the body. And it'll wrap itself around it and it'll put it inside of a vesicle. It's a type of endocytosis where it basically eats another cell, which is called phagocytosis. The other type of endocytosis is if you're dealing with something that is more liquid-like or ions, little droplets or ions. This is not cell eating, it's almost like cell drinking. And so this is called penocytosis. It's a similar process, but because you're dealing with a smaller molecule or individual molecules or fluids, then it's called penocytosis rather than phagocytosis. There's also something called receptor-mediated endocytosis. This is similar to phagocytosis, but in this case, it is stimulated by receptors. So in this case, you've got some kind of cell. It's got little receptors, protein receptors on the membrane. Whenever that ligand, remember that definition, ligand, that little chemical that binds to the receptor, whenever it binds, it stimulates the cell to start the process of endocytosis. So the only difference here really is that you have receptors that start the process of endocytosis. In the next video, we'll go over section 4, which is going to help go through the different cell organelles that you find inside of the cell.